Hi students, this is Mr. Eckert and we're going to go through targets 3 and 4. 3 says that you should be able to identify the different cell parts and on the test I'll give you a drawing that's unlabeled of course and you'll have to label the different cell parts. And then target 4 says that you should be able to describe the function of each of those parts. So we're going to go through both of these targets kind of at the same time. So first, a part of the cell that I'm sure you're very familiar with is the nucleus. Again, this is not something that all cells have, but something that all eukaryote cells have. They have a nucleus and it acts like the control center or the brain, regulating everything that goes on inside the cell. So you can see the nucleus here in this cell and in this plant cell down here. So the nucleus, there's 3 other parts that you should be aware of for the nucleus. There's the nuclear membrane, that's just the outer layer of the nucleus, and that separates it from the rest of the cell. It also has these holes or pores in it through which substances can move in and out of the nucleus. Then there's the nucleolus, and that's an inner section of the nucleus that is clearly defined as being different. And in the nucleolus, the ribosomes, which you might remember they make proteins, they're assembled there. And then finally the nucleoplasm, just like the cell has a cytoplasm, clear kind of goopy stuff in which everything floats, the nucleus has a nucleoplasm in which everything inside the nucleus kind of floats and moves around in. So in the back of your guided notes you should have this diagram as well as a plant cell diagram, and you can label the outer nuclear membrane, the nucleolus, the nucleoplasm, the nuclear pores, and all of those, well then you have the inner nuclear membrane, all of those make up the nucleus. In the plant cell you've got the nucleus, the nucleolus, the nuclear membrane, and nuclear pores. So the nucleus, this structure here. Chloroplasts, these are found only in plant cells. It says some algae, but nearly all algae have chloroplasts. And chloroplasts is where photosynthesis happens. So you in the lab will have an opportunity to see this plant live, and you might even see, these are the little chloroplasts, you might even see them actually moving around inside the cell. So the green pigmentation that plants have, that's because of the chloroplasts. So obviously you're not going to see these in your animal cell. In the back of your notes, the plant cell, here's chloroplast, here's one right here, here's another one, here's another one. Now if you're going to confuse this with anything in the cell, it would be these. These are mitochondria, and you notice they have kind of a folded inner membrane, whereas the chloroplasts are almost always drawn with these stacks inside of them. So that's one way you'll be able to tell them apart. Speaking of the mitochondria, mitochondrion is singular as it says, so mitochondria is plural. And this is where the cell really makes most all of its usable energy, or ATP. ATP is the molecule of usable energy. So cellular respiration is the name of the chemical reaction that happens inside of mitochondria. And so, as it says, you may remember from middle school, it's called the powerhouse of the cell, or sometimes it's called the mighty mitochondria, because it's making usable energy. So in your animal cell, you'll see the mitochondria here, and here's another one. Now, of course, there would be many more than just 2, but for simplicity's sake, they've only drawn 2. And in the plant cell, you've got a mitochondria here, here, and here. Ribosomes, remember that ribosomes make proteins. Proteins are the building blocks of living things. So sometimes you will find these attached to a structure called the endoplasmic reticulum, and ribosomes are almost always drawn as little dots. They're very small structures. Or there's free ribosomes that float around in the cytoplasm, and there'd be tens and tens of thousands of these in a real cell. So in your animal cell, you've got ribosomes on the ER, the endoplasmic reticulum here, and you can see some more here. and then you've got some free ribosomes here. They've only drawn 3, but there would be many more than that. In the plant cell, you've got some rough endoplasmic reticulum here with you can see the ribosomes on it, and maybe we have some other ones there. A cell wall. Cell walls are only found in plant cells and in bacterial cells and in fungus cells. Now that's omitted here from the notes, but you might want to add that in. So found in plants, prokaryote, and fungi. So you can see here's an electron microscope photograph of a cell wall, and you can see it provides a lot more structure and rigidity. It's thicker than a cell membrane, so it provides some protection for the cell. So on your plant cell, this section right here is the cell wall for this main cell. And then sometimes students get confused about this, but this plant cell is shown in relation to some other plant cells. So you can see these are the cell walls of adjacent plant cells. And they have pores as well to allow substances to move in and out. Cell membrane is also called the plasma membrane. And so you can see a drawing of it, a cartoon drawing of it down here. And you can, if you look closely, you can see it's really 2 layers. It's a layer on top, and it looks like these circles. If you look really close, you can see these little tails. They almost remind me of balloons. And then there's a layer on the bottom. So these are called phospholipids. And they have one part that is hydrophilic. It actually likes water, and that's the part that looks like a balloon. And then the part in the middle where the tails are, those are called hydrophobic. Hydro, of course, means water. Philic means love. Phobic means a fear, as you know. So obviously it's not that they're afraid of water, but the inner part actually repels water, whereas this outer part that looks like a balloon would allow water to pass through. So they are selectively permeable. If you think about what that means, if something's permeable, it allows pretty much all substances to move through. If it's impermeable, things can't move through it. So selectively permeable means that it's going to allow some things to move through and prevent other things from passing through. So then there are some proteins that are these big fat things that you see here and things that look like strings coming out. These are different kinds of proteins that some of them are marker proteins and help the cell to recognize other cells around it. Some of these are receptor proteins, and molecules will bind to those, and then the cell will decide on whether or not that's something that should be allowed in or not allowed in. And some of these are transport proteins that actually help substances to move across the cell membrane. So on your animal cell, this outer layer is the cell membrane, and on the plant cell you can just barely see this thin inner membrane. So the cell membrane is always on the interior surface of the cell wall in relation to the cell itself. Endoplasmic reticulum, there's 2 kinds, smooth endoplasmic reticulum and rough. Now remember, rough has the ribosomes attached on the outside, whereas the smooth does not. The endoplasmic reticulum, its main job is to help move proteins and other substances in and throughout the cell. It actually connects as you can see in this drawing, the nucleus. It connects to its outer membrane and goes all the way out to the cell membrane. You don't really see that from this drawing, but you do see its connection to the nucleus. So on your animal cell, here's the rough ER again, and it would actually be connected as it shows this one, connecting to the nucleus. And here's some smooth ER down here. Now again, sometimes students will confuse it with this structure here, which is known as a Golgi apparatus. Golgi apparatus is a packaging. It modifies different proteins and other substances and packages them. So they almost always draw it with these little packages, which are called vesicles, and that's one way you can tell the Golgi apparatus apart from smoother rough ER. In your plant cell, again, here we have a rough ER right here on this side of the nucleus, And here's some smooth ER without the ribosomes over here on this side. Golgi apparatus, we said it consists of stacks, different stacks of membranes, and they receive and modify substances and then move those substances to other parts of the cell or all the way out of the cell. So you can think of it as a packaging and distribution center. Sometimes students will relate this to like a post office, which is packaging and moving things. And this down here was taken from an electron microscope photograph, but of course it's a computerized image of what the Golgi might look like. So in your animal cell, again, here it is down here. And in your plant cell, we've got a Golgi apparatus right here. And here you see those vesicles that I mentioned, those are the actual packages that it's making. Okay? Lysosomes. These are sometimes called suicide sacs because these are filled with digestive enzymes. You can think of them almost like acids that break substances down. So these come from the Golgi apparatus. It actually produces them. And these would be scattered throughout the cell. And their function is to break different substances down. It could be something foreign that shouldn't be in there, like bacteria. It could be organelles that have gotten old and are no longer functioning properly, so they're broken down and then parts are recycled. So it's a recycling and digestion location, or that's its main function. So in your animal cell, now these are also kind of drawn as circles, bigger than ribosomes usually. So here's a lysosome here, here's probably another one right here. In the plant cell, you've got one here, and looks like here, here, and here. So I think we're going to stop at this point where this section is rather lengthy, and so we're going to divide it into two parts. This will be part 1, and you'll pick up part 2 and finish out the rest of your notes on another video.